I'm Robert LeBoyer, Senior Biotechnology Analyst at Noble Capital. With us here today for our first presentation is Dr. John Allen, CEO and President of CervoMed, a clinical stage company developing central nervous system focused therapeutics for patients with a range of debilitating neurodegenerative diseases. Go ahead, Dr. Allen.
Thank you, Robert, and thank you to Noble for inviting us to the conference. Thank you to everyone who's listening in. Good morning, everyone. I'm really excited and thrilled to be able to introduce, for particularly the people who are new to CervoMed, our company based in Boston that's a leader in developing a therapy in particular for dementia with Lewy bodies. I would urge you to go and for a fuller description of our company, including the risk factors in the company, to go through our various filings with the SEC, including our 10K, which was filed recently on March 29th. So CervoMed was launched as a company named CervoMed in last August, following a merger between a private company, EIP Pharma, which I had founded and had been CEO of for the last few years, and a merger with Diffusion, a public company, Diffusion Pharmaceuticals. We're headquartered in Boston. Our lead program is an oral drug named Neflamapimod for the treatment of dementia with Lewy bodies. So with the merger, we have put together really a world-class leadership team with the deep experience in drug development, taking drugs all the way through to the market, and a board of directors that's commensurate with that team and is, again, very much on the leading edge in our industry. Along with that drug development experience, very deep network and understanding of the biotech healthcare investment community. On our board, I'll just point out Joshua Boger, who came onto the board and became chair in early February. He's a very well-known in our industry as a founder and former CEO for 20 years at Vertex Pharmaceuticals. One comment on the financial overview, and then I'll go to really the science and the clinical data, and the opportunity around our drug in dementia with Lewy bodies. We did just very recently complete a PIPE with leading healthcare investors. This was completed on April 1st, a couple of weeks ago. This was led by RA Capital, a very well-known leading healthcare-focused investment firm. With this, we received $50 million, which funds us fully through to the end of next year, 2025, including the startup phase of the planned phase 3 trial in dementia with Lewy bodies that we are targeting starting in the middle of next year. There's an additional up to $100 million that would come from the exercise of warrants, which would have to be completed with success in our phase 2b trial within 6 months of the data announcement, which we anticipate this point in the fourth quarter of this year. That the warrant exercise price has 100% premium, or it's double what the upfront price was for the $50 million. Very quickly, at a high level, what I'll be talking about, again, introducing you to the data and the opportunity with our drug in dementia with Lewy bodies, which is a very major neurologic indication, high unmet medical need, and with it, high commercial opportunity. We are the leader here for being the first to market with a drug into treatment of dementia with Lewy bodies. We're the only ones with positive phase 2 data, and it's data that's been published in major journals and high-impact journals. We're the only ones in a phase 2b clinical trial, which is actively enrolling today. We anticipate completing enrollment within this quarter, the second quarter, and data readout in the fourth quarter of this year. The investment opportunity in the near term is very much around this phase 2b clinical study, and much of what I'll be talking to you about is why we believe our probability of success in this clinical trial is high, and I'll give you the reasons for that and the data out of the data that we have to this date, as well, given the nature of our dementia with Lewy bodies, how our ACT drug works, the benefit we provide to patients, that we have with this phase 2b study, we will substantially de-risk phase 3, and with that create substantial value in the drug and in the company with success in this phase 2b clinical trial. So dementia with Lewy bodies, again, third most common degenerative disease of the brain after Alzheimer's, and there's Alzheimer's, there's Parkinson's, and then there's DLB. It is a very distinct dementia from Alzheimer's disease. In the first point is that it's not a memory disorder, it's more deficits in attention and something called executive function, which is things like judgment, reasoning, problem solving. And then for a diagnosis, and what invariably goes with true DLB is two of the following: fluctuating attention, visual hallucinations, a very specific sleep disorder, as well as certain of the motor deficits of Parkinson's disease, and that's called Parkinsonism. If you have two or more of these associated features, or what are called core clinical criteria, then you have DLB. That's not AD, that's not frontotemporal dementia, that's DLB. Because of these features, particularly the combination of cognitive impairment or the dementia with motor deficits, the impact on patients in terms of quality of life, caregiver burden, high healthcare costs, all of those combine to make it, when you stage match, in particular in the early stages, substantially worse than Alzheimer's disease. That high unmet medical need, combined with this being a specialty disease in that once diagnosis is managed by neurologists, is what makes this a significant commercial opportunity. So from a clinical development standpoint and from a scientific standpoint, while the disease is actually terrible and has much more impact on patients and actually progresses faster than Alzheimer's disease, at the same time, it's actually in other respects not as bad as Alzheimer's and not as aggressive in terms of actually apprehensible in terms of treatment. And the reason for that is that in the early and middle stages of the disease, there's actually less frank neuronal death and loss compared to Alzheimer's disease. In early stage DLB, which makes up as much as about half of all patients with dementia with Lewy bodies, it's actually a functional deficit. The neurons are sick, they're not functioning well, there's significant symptoms because of that, but they're rescuable. If you have the right mechanism that targets the right set of nerve cells in the right part of the brain, you can actually heal, make the neurons, the nerve cells function better, and actually reverse clinical progression in that actually restore function and make patients better, not just as we see with the Alzheimer's disease treatments, even the best ones, slowing future decline without giving anything back in terms of the near term. What that does from a drug development standpoint, it gets one out of the trap of what Alzheimer's disease or the chronic neurodegenerative disease drug development challenges, which is that you can't show this improvement. Rather, you depend on long-term clinical trials and large trials to get enough placebo progression to be able to show a difference with drug treatment. And even when you're successful, the absolute difference from not treating or being on placebo is always small. The bigger challenge from a drug development standpoint is that you can't get efficacy in a phase 2 clinic, definitively demonstrate efficacy in a phase 2 trial, because at the end of 6 months, and phase 2 trials are typically 3 to 6 months in duration, this difference is too small to pick up. You can see, you can see biomarker effects and a trend, but you always go into phase 3 with most of your risk in front of you. In contrast, in a disease like DLB, an early stage DLB, you can see this improvement. You can demonstrate efficacy in phase 2. And more importantly, perhaps, your phase 3 trial can be 6 months in duration and more or less be the same as your phase 2 trial. And you can go to the market with that. We know that in talking to the FDA, if you can show a clinical meaningful difference at 6 months, you can get approval on that basis. And your phase 3 trial, in being a replication of what phase 2 is, if you have success in phase 2, you will have taken the risk and substantially most of the risk off of the program before even going into phase 3. Now, that reversible component of disease shows up in what's called the pure DLB population. It's a diagnosis effectively of exclusion. The more advanced patients who have more significant neuronal loss, invariably with that, they have what's called biomarker evidence of Alzheimer's disease. So they'll be amyloid plaque positive on a PET scan. They'll have CSF markers, and they'll have blood markers, in particular elevations of phosphorylated tau, this protein, or for example, a p-tau 181, which is what we're using. If you have that elevation, you have what's called DLB-AD. You can develop a drug here, but you'd have to develop it along the lines of what you do in Alzheimer's disease, long duration trial. Most of your risk still remains when you go into phase 3. In contrast, we're focused on pure DLB. We have data in this population in particular that looks great, looks very good. It has a reversible component. We believe we're going to be able to demonstrate efficacy in phase 2. We can go to the market on a 6-month treatment trial, and therefore that result will de-risk phase 3. So the drug is an oral drug, targets a very specific mechanism. We licensed it from Vertex Pharmaceuticals, who had been developing it for diseases outside the brain. It was shelved. It was actually a decision I made when I was there at the time in previously, because brain concentrations are actually higher than outside of the brain. That doesn't work for rheumatoid arthritis. It stayed there until new science came around that made it interesting to develop this mechanism in CNS disorders, for neurologic disorders. And it has this profile that we can give it at low doses, have minimal systemic effects outside of the brain, so minimizing risks in terms of side effects, while retaining a very specific effect in the brain. We've shown this in multiple ways, both in animals and the clinic now. But from a safety standpoint, we have significant amounts of data, both in animals and in the clinic, that we believe, because of this profile in particular, from a safety standpoint, we're in great shape. We're in good shape. So mechanistically, I won't go into the science. We act on a very specific part of the brain, on the basal forebrain, on neurons called cholinergic neurons. These are neurons that produce a neurotransmitter, acetylcholine, that's involved in regulating a variety of cognitive and motor tasks. We work with some of the leading neuroscientists to demonstrate our drug through reducing our primary mechanistic target activity of p38 alpha, reduce a series of downstream events, and eventually reverse the disease in the cholinergic system in the basal forebrain. We've published that data along with our primary clinical data from the phase 2A study in Nature Communications. This was published in September of 22, where we showed from the clinical standpoint in a 16-week clinical trial against placebo significant effects on the standard way of assessing overall dementia severity, as well as evaluating effects on walking ability, on gait, We also saw effects on cognitive results that was dose dependent. In particular, we saw that the higher of 2 doses in the study is the reason why we are progressing with this higher dose level in 40 milligrams 3 times a day. We have a second publication of the study that was published in October of last year, where we went back to this concept of pure dementia with Lewy bodies, and using a blood test, identified those patients in that study who had DLB-AD and did an analysis focused on this population, the pure DLB population. These are the overall, again, study results. In the overall study population, significant effects on multiple clinical endpoints, but in that pure DLB patient population, the magnitude of effect roughly doubles. If using a standard measure of magnitude, Cohen's d effect size, we also see effects on memory, which we did not see in the overall patient population. So very consistent, somewhat unprecedented levels of activity for diseases like any dementia indication. And we see significant effects against placebo with relatively small patient numbers. We also recently presented data that in that pure DLB population, there's a significant biomarker effect on a biomarker of the underlying disease process called GFAP. And we could correlate that to a clinical result in terms of dementia severity. Again, somewhat unprecedented to be able to see both a biomarker effect and one you can correlate to clinical results, which again, we believe has to do with we're matching mechanism to disease, as well as we're in this treatable patient population in that pure DLB patient population. So that phase 2A study was an exploratory study, is phase 2A. We're not claiming efficacy yet. We need to confirm that in something called a hypothesis testing trial, which this phase 2B trial is, where we have, it's a somewhat of a replication of the phase 2A study in that it's 16-week placebo control, but it's higher patient numbers, 80 patients per group. Primary endpoint of that dementia rating scale, CDR sum of boxes. We're excluding those patients with DLB-AD. This trial is actively enrolling. Again, we anticipate fully enrolling the study within this quarter, and this 16-week endpoint, the primary efficacy analysis, the data readout in the fourth quarter of this year. In terms of probability of success, I think the easiest way to think about it is that we saw significant effects at this dose level with 10 to 15 patients per arm versus placebo. We're now at 80 patients per group. We think it's for that reason we believe that we have a really good shot in this trial, and it should read out positively later on this year. And again, the phase 3 study, we know based on discussions with the FDA, will be, again, a replication. It'll be a 24-week study. CDR sum of boxes, based on our discussions, would be an approvable endpoint. But because it's very similar, only differences are a little bit longer for safety. To get enough safety data, it'll be bigger. All of those things work in our favor in terms of demonstrating efficacy. For those reasons, with success in phase 2B, we believe that we will have de-risked phase 3, and the cost will be nowhere near what a study in Alzheimer's disease would be. All around, the net asset value should be substantial with positive success in phase 2B. Through the year, with this financing, we have ability to then look at additional indications as well. We'll communicate around this as we go through the year, even ahead of the phase 2B result. And it's one of the reasons for these are potential catalysts in terms of how our profile of the company and how our company is valued. And so and I'll conclude with that. Again, this is an exciting year for us, which we already the first quarter with the financing, with bringing Joshua Boger on board. It's been a great first quarter, but there's more ahead through the year culminating with the data readout in dementia with Lewy bodies. And I'll stop there.
Thank you, Dr. Allen. That was a very interesting presentation. And before I open up the floor for questions, I just wanted to ask one or two things about the clinical trial. And it sounds like you're doing very careful science to characterize the drug so you know its effects, and you can design all of these subsequent clinical trials to prove your hypothesis, which, like you said, that's reducing the risk and raising the probability of success. But in terms of the design, how is the drug administered?
It's an oral drug. It's
Okay,
very,
so
very straightforward. Our compliance adherence in all of our prior clinical trials has been great. Again, just to be clear, that I think that we would say that all of the work we've done in AD, previous in dementia with Lewy bodies, where we've gotten really good understanding of drug mechanism, target engagement, we've seen MRI effects, spinal fluid effects, combined with the phase 2A data, I think we've learned about as much about a drug as anyone has done in diseases like AD and DLB. I think we have already substantially reduced the risk. I couldn't go into as much data as you could. I would encourage the people who are listening to go onto our website. We've been very transparent. We've aggressively published. We have an enormous amount of information out there. We truly do believe the drug is working in DLB. It's a matter of running out this trial in phase 2B and showing that. But that risk we've already substantially reduced, even going into the phase 2B trial in this program. And I hope that 25 minutes or 20 minutes, difficult to convey all of that. As I said, it's very much on our website and the publications that we've had out there.
Yeah, and having read some of those publications, I would agree with you. And one of the things that jumps out about your clinical trial design is that the 2B and the phase 3 that you're talking about have the same design as the 2A, and you're proving it with larger numbers for statistical power and running it longer for safety requirements for approval. So you've eliminated most of the variables that lead to clinical risk and clinical trial failures. So
absolutely correct. And it is again, I don't think this is magic. It is a being right place at the right time with the right drug, where a lot of what's come together in the field in AD and DLB of using these biomarkers to identify very specific patients where you have the driving mechanism that you are targeting is actually, you can match your drug to patient, and this pure DLB patient population we're doing, that's what's allowed us to really I think make a difference and really truly understanding and getting an optimal effect in the drugs. It's the way of the future in all of these diseases, but for our drug in DLB, I think the future is today. It's just this being able to do that, and it's very exciting for us. And everyone we talk to in the field in DLB, they're just extremely pleased that in a disease that a few years back just seemed enigmatic, but it's all come together both in disease understanding and DLB. It's been just breathtaking advances in the last few years in terms of understanding DLB as a disease. And we have the right drug for this disease, and we've been able to connect into that.
Yeah, and as an orally available drug, it shows effects very quickly. And some of the Alzheimer's drugs that have come out require IV infusions for 18 months before they see a difference. Your drug actually reverses some of the damage and restores function instead of just slowing the deterioration. So I think what you described is something that's easy for patients to take as an oral, and they'll see effects very quickly and stay on it. One other question would be, having looked at the mechanism and read some of the papers, is this drug something that can be applied for other CNS degenerative diseases?
Yes, and I would say that more broadly, it can be applied for a range of age-related disorders. And the reason the age matters is that inherently with age, there's a deterioration of the function of that part of the brain that I talked about, the basal forebrain cholinergic system. And so there's a whole range of diseases where that system actually is playing a substantive, if not the major role. And so one of them is actually the recovery mechanisms after a stroke. So this is not neuroprotection. It's not giving it acutely while you're having the stroke to try to reduce the size of the stroke. But that cholinergic system is absolutely critical in recovering function in the weeks and months after a stroke, particularly around motor function, like things like being able to walk, or using, a knife and fork to be able to eat. These are things that are critical, obviously, to a stroke patient. And the cholinergic system, because of the residual inflammation in the weeks after a stroke, can shut that system down and have negative effects on that. And we have great preclinical data, for example, where we can reverse that and improve motor recovery. So that's an example. And there are a number of other diseases like certain forms of frontotemporal dementia that could be very much on the table as well. And as I said, that you will, stay tuned. We have not any announced plans at this point. But with the financing, with the PIPE that we completed, we have the ability to now actually start looking into some of those as well. And we will be communicating around that through the
Great. Okay, and just one final question. Could you just repeat some of those milestones in the next several months that we can look forward to?
Well, the major one is around the phase 2b clinical trial, obviously, and there again, it's to complete enrollment within the second quarter, so this quarter, in the coming weeks, by the end of June. And then the data readout from that phase 2b trial will be approximately 6 months after that. And so within the fourth quarter. And we're working very hard on the enrollment, but then also to get the data and then really have everything in place with data we have the ability now, I think with the financing, to run with that. Go to the FDA, get the phase 3 design finalized with a target of starting phase 3 in the middle of next year.
Great. Well, thank you very much. We're out of time. I wish we could continue, but I'd like to thank you for joining us today, and we'll be looking forward to those milestones.
It's great. Thank you again for the invitation. It's always great to speak with you, Robert. And thank you to everyone watching and listening in today.
Thank you.
It's to complete enrollment within the second quarter, so this quarter, in the coming weeks, by the end of June. And then the data readout from that phase 2b trial will be approximately 6 months after that. And so within the fourth quarter. And we're working very hard on the enrollment, but then also to get the data and then really have everything in place with data we have the ability now, I think with the financing, to run with that. Go to the FDA, get the phase 3 design finalized with a target of starting phase 3 in the middle of next year.
Great. Well, thank you very much. We're out of time. I wish we could continue, but I'd like to thank you for joining us today, and we'll be looking forward to those milestones.
It's great. Thank you again for the invitation. It's always great to speak with you, Robert. And thank you
Thanks
to
again.
everyone watching and listening in today.
Thank you.